Our bodies run on millions of microscopic machines called proteins. They handle virtually every physical process inside of you, from digesting food to neutralizing incoming viruses. Their ability to function depends entirely on this specific transition. A protein starts as a linear chain of amino acids, but it quickly folds into a complex three-dimensional structure. In biology, that shape dictates exactly what the protein can do. For decades, identifying those shapes required painstaking physical labor. It took massive, complex hardware, like this Bruker 400 NMR spectrometer, to manually analyze molecular structures. It was a slow, expensive process. Then DeepMind developed AlphaFold. Instead of relying on years of physical lab tests, this deep learning algorithm successfully predicted how these proteins fold using computational models, mapping static structures down to the atomic level. This shift moved structural biology from the laboratory bench to the GPU. By making these shapes predictable through data, researchers are now free to investigate how these molecules behave in a living system. But AlphaFold's results have a specific limitation. The structures it generates are frozen. They are snapshots of a molecule at rest. In reality, functional proteins are in constant motion. They twitch, morph, and undergo conformational changes to execute their tasks, rapidly shifting their physical configuration to lock into other molecules. A static blueprint only tells part of the story. To see how life actually functions, artificial intelligence must learn to simulate these molecules as they move. Researchers are now training next-generation AI models to integrate dynamic simulations over time, attempting to capture these rapid changes frame by frame. By watching these structures move in real time, scientists can see the exact physical mechanisms of enzymes at work, or spot the precise moment a protein folds incorrectly and triggers a disease. Proteins rarely operate in isolation. As you can see in this model, they interact with a vast ecosystem of non-protein molecules, locking together with RNA strands, lipids, and carbohydrates. New AI systems are expanding their predictive reach to map these interactions. They are learning to model the structures of these other essential biomolecules to see how they fit into the wider cellular environment. Charting these non-proteins changes our perspective. Instead of looking at individual components, we are beginning to view the cell as a single, fully interconnected machine. That machine produces massive, overlapping datasets. Biologists call this multi-omics data, which includes the entire catalog of a cell's genes, its proteins, and its metabolites. The sheer number of interactions happening across those layers simultaneously is staggering. It is a level of complexity that is impossible for the human brain to process manually. AI bridges these layers. It takes disparate datasets, visualized here as distinct data planes, and stacks them, drawing vertical connections to form a comprehensive network. By mapping this, researchers can trace a faulty connection from the top down to its molecular root, pinpointing disease. These algorithms also assist in the physical lab. Machine learning is now used to automate the fine-tuning of delicate experimental equipment, like cryo-electron microscopes, increasing the resolution of the data we collect. AI has become a necessary research partner, closing the loop between digital simulations and physical laboratory realities. Scaling that cellular network up to the level of a patient introduces a new level of drug discovery. Before a new medication is even synthesized in a lab, AI can simulate the physical interaction between a proposed drug molecule and its target protein to see if they match. The goal is to take those molecular simulations and match them against a patient's unique genetic profile. This allows doctors to predict how well a drug will work and identify potential side effects before the patient ever takes a dose. Treatments can be developed in a fraction of the time and customized to the individual, moving us away from trial-and-error medicine. But as our ability to simulate human biology improves, significant security risks begin to emerge. Personalized medicine requires the storage of massive databases containing sensitive human genetic information, which creates a target for data theft and misuse. There is also a dual-use threat. The same software that can design a molecule to repair a cellular receptor can be repurposed to design a molecule that permanently disables one. When an algorithm understands the precise mechanical fit of human biology, it can be used to engineer both cures and toxins. This makes robust regulatory frameworks and international cybersecurity cooperation essential to prevent the development of bioweapons. Biologists and computer scientists must work with ethicists to ensure these tools are developed responsibly and to identify potential biases in the algorithms. AlphaFold provided the initial maps of these molecular structures. The next generation of biological AI gives us the power to re-engineer them.